Well, all right, BIO1030 class. Today we're going to dive into the spinal cord and how the central nervous system is protected. Okay, so let's dive right into it. Just as kind of a fun question, how many neurons are there in the human spinal cord? So spinal cord, not the brain, but the spinal cord. Okay, we've got about 1 billion neurons in the spinal cord. Okay, how many in the brain? 100 billion in the brain, 1 billion in the spinal cord, brain. Okay, so lots of neurons here, nothing even close to what we have in the brain though. Okay, we're not going to show this video, it's just a brief overview of the central nervous system, just kind of stuff that we already know. Okay, so let's look at the spinal cord, the makeup of the spinal cord, what we call the cross-sectional anatomy. Okay, so if we just slice the spinal cord and then look at it from the top, what do we see? This is what we see. Okay, so we have these just kind of indentations called sulci. So we have the dorsal median sulcus and the ventral median sulcus. Okay, just to get yourself familiar with this again, obviously the dorsal side, the ventral side over here, that's that dorsal root ganglion that we've talked about before. These are the roots and the rami, like we've talked about before. Okay, so let's talk a little bit about the spinal cord itself. So we've talked about these, right? We've talked about these, what we call the spinal nerves. But what about the spinal cord itself? Okay, so we've got two portions, the gray matter and the white matter. So the gray matter is the stuff in the middle, almost kind of looks like a butterfly, right? And the butterfly wings here, first of all, we have this first portion, which is the gray matter. And the gray matter is gray because it's made up of cell bodies, okay, somas, and then all the synapses of other neurons, okay? That looks more gray compared to the white matter, okay? And we can see how this comes out. So we see the posterior root, sensory part, coming out to the sensory innervations here, and then the ventral root coming out to motor innervations there. Okay, so let's look at each of these little portions because they're each a little bit different. So the little tips on the dorsal side, these are known as the dorsal horn. This is obviously the afferent side. So it receives afferent information and transmits to the brain, right? Because those afferent neurons are coming in here, and then it's going to go straight up to the brain from there, right? Remember, these are what these dorsal root ganglion cells look like. The cell body, oops, the cell body would be right here, right? And then we've got the neuron going, or axons going both directions, this way going up to the brain, this way going out to the receptor that actually receives the information. Okay, and then the other side, the ventral horn is what that's called. These are specifically cell bodies of somatic motor neurons, right? Because we know that ventral side is efferent, so it makes sense that these are motor neurons, and specifically these are the somatic ones. Okay, signal starts way up in the primary cortex of the brain. Okay, we'll talk about that primary cortex, comes all the way down, crosses over at some point, comes out through the ventral horn and out to skeletal muscle. Okay, and then we got these little lateral tips. Okay, the lateral horn, these are cell bodies of autonomic neurons. Okay, for the abdominal organ. These are only really present in the middle of the spine, T1 to L2. Okay, and these are more specifically for the abdominal organ. So these are the neurons that come out and control our digestion, our breathing, or things like that, or not breathing, but our digestion, all throughout our abdominal cavity. Okay, what about the white matter? So not the butterfly part in the middle, but the parts on the outside. Okay, so that white matter is white because it's myelinated axons. And what's myelin made out of? It's made out of lipid, right? And lipids are typically white. Okay, so cross-sectional anatomy recap here. Okay, so in general, what is gray matter? Well, cell bodies. What's white matter? Axons. Okay, the efferent comes out of the ventral horn, afferent comes into the dorsal horn, and the autonomic goes out of the lateral horn. Okay, all right, so just the general gross anatomy of the spinal cord. It extends all the way from the foramen magnum, which is that hole, right, in the bottom of the skull, all the way down to L2. After that, it's something different. We're going to talk about what it's called when it gets down there, but the true spinal cord is just that part right there. Okay, so we have a couple enlargements in the cervical portion, we have a cervical enlargement, a lumbar enlargement, and then after L2, we call it the cauda equina, which means the horse's tail. Because if you've seen a horse's tail, it kind of comes down and it spreads out. That's kind of how the spinal cord becomes at that point. It's not called the spinal cord, it's called the cauda equina. And you can see that there, how it's kind of spread out. It looks like these long, thick bands. All right, so let's talk a little bit of clinical application here of the spinal cord. We're gonna talk about these different motors here in a second, but the dura mater is this tough connective tissue layer on the outside of the central nervous system. Okay, and then there's a ligament out here, external to that, and then it gets to the vertebrae. Okay, and down inside there is where the spinal cord is, right? So it's in here. Okay, so there's a couple different procedures. One known as an epidural, right? If you've had an older sister or aunt or somebody you've or maybe your mom, if you have a lot of younger siblings, been a part of and been involved in their delivery process of a baby, then you might have been a part of conversations where you've heard of people say, I got an epidural, right? Or I'm kind of considering whether or not I want to get an epidural, because an epidural is pretty much you put numbing medication into the spinal cord, at least to the outside of it, and that numbs up that the whole bottom half of the lady's body, so she doesn't feel as much of the pain from the delivery. Okay, very common procedure, very wonderful procedure to let the woman enjoy childbirth. Okay, so we've got an epidural, means that it's outside of the dura mater. Okay, so when they stick that needle in, it goes through that connective tissue, goes through the muscle there, goes through that ligament layer, and right when it gets through the ligament layer, there's a little space there called the epidural space, and that's where they're supposed to stop. Okay, they're not supposed to push any further into the spinal cord, so it actually stays outside of the spinal cord. Okay, so that's where the fluid's inserted. Another type of procedure is called a spinal tap. And a spinal tap, this is different, right? Spinal tap, you actually get all the way in, you push through that ligament layer, but then you also push through that dura mater. You get into where the cerebral spinal fluid is, and you draw some fluid out. That can be used to test for certain things inside of the central nervous system. Okay, and this is all done in the cauda equina because there's more space in there. Right, there's more room. Okay, if we did it up higher up in the spinal cord, then you'd be more likely to poke into a nerve when you went in, instead of up, down, lower in the cauda equina where there's some more room, and hopefully you won't damage something when you get in. Okay, so let's do a little bit of labeling here. These different portions, we've kind of already done this, right? So this is just kind of review. Dorsal root is where the afferent neurons are. Dorsal ramus, talked about these. Ventral root, efferent neurons. And then lastly, or in the dorsal root ganglion, and lastly, the ventral ramus. Okay, so that's just a kind of a recap of what we've talked about previously, and now it makes a little bit more sense. Work out the cell bodies of the afferent neurons there, right? Cell bodies of the efferent neurons here, cell bodies of some efferent but autonomic neurons there. All right, let's talk about the protection of the spinal cord, or the CNS. How do we protect it? Obviously, the skull is incredibly important for the brain. So the brain's protected by the skull. Spinal cord is protected by those vertebrae, right? Those vertebrae have a nice hole in them, but it's completely surrounded by a bone. Nice hole for that spinal cord to pass through. But then you can also see this fat layer in here, right? See that fat right there? That is also very protective of the spinal cord as well. And then both of them are protected by meninges. We'll talk about what those are in one sec. Cerebral spinal fluid, and then the blood-brain barrier. Okay, so this is just kind of showing how well those vertebrae protect the CNS, right? So spinal nerves exit via intervertebral foramen, right? So these little holes right there, passing through those little bony canals, very important for protecting those spinal nerves. All right, let's talk about the meninges. The meninges, again, the first one's called the dura mater. Excuse me, sorry. I tried to pause it, but didn't get to it in time. Okay, protection of the CNS, the meninges, the dura mater is the thickest one. It actually means, in Latin, tough mother. Okay, it's thick, it's tough mother. Okay, so the dura mater is typically actually 2 layers in the brain. but they're fused, okay? And we can see that. So here is the skin on top of the scalp. Here's some periosteum, right? And then the bone of the skull, okay? And then underneath that, we've got 2 layers of dura mater, okay? We've got the periosteal layer and then the meningeal layer. So all of this is dura mater. So big, thick, tough connective tissue. That outer layer sticks to the skull. The inner layer separates the dural sinuses. Okay, so there's the dura mater in the spinal cord. It's only a single layer in the spinal cord instead of that 2, because there's something else between the bone and it. There's fat in the fat layer. We don't have that in the brain. Okay, the next layer down is called the arachnoid mater. Okay, and we can see that here. Okay, and then there's this space, kind of a diffusion type space. And then the little one that's actually touching the brain tissue itself is called the pia mater. Okay, so when we talk about intracranial bleeds or bleeds in the brain because of traumatic brain injury, they're typically named by where the blood is in relation to the different meninges. Okay, so if we have something called an epidural bleed, that's on the outside of the dura mater, right? So it's outside of all the meninges, that would be here. Okay, so it's between the skull and the dura mater. This can be dangerous because it can push in right here, right? Okay, and we've got the subdural hematoma or subdural bleed. Okay, so it's underneath the dura mater. So you can see that blood getting in there, fills that space, can also be very dangerous. Okay, we have what's called a subarachnoid bleed. Okay, which actually gets down in here a little bit. So it's just between the pia mater and the arachnoid mater. And then lastly, if we get down below the pia mater, then that's known as an intracerebral bleed or parenchymal bleed. And that will be all the way down in the brain tissue. Okay, so just need to be familiar with what those different terms mean. Okay, so these meninges can become infected, and that's when we get what's called meningitis. Okay, meningitis, we do have a vaccine for it now, but it protects specifically against bacterial meningitis. Viral meningitis is also very dangerous. Okay, this is when this gets, the meninges actually get infected. If that infection spreads into the brain, it can cause permanent brain damage as well as death. Okay, the brain is also, or the whole central nervous system is also protected by cerebrospinal fluid. The brain is actually kind of floating on cerebrospinal fluid, right? So there's really fluid all the way around the brain, okay? And then there's fluid on both sides of the spinal cord as well as in the central canal going right through the middle, okay? And remember those ependymal cells, which were one of the glial cells that we talked about, those are the ones that are making the cerebrospinal fluid. So they're pulling fluid out of the blood and then releasing that fluid into the cavities of the brain. Okay, we have some big cavities in the brain called ventricles. Got some here, got some here. And remember those cilia are beating and circulating that fluid all around. Okay, so cerebral spinal fluid is very similar to the plasma part of our blood. So that's the water portion of our blood, okay, but not quite as many proteins as we have in our blood. It's only about 150 mils of fluid in an adult, so small amount of fluid, but this stuff is constantly turned over. Okay, so we form about 500 mils of this a day, and so that means we reabsorb about 500 mils of this a day. So it's constantly being replaced. About every 8 hours, or about 3 times a day, we completely have new cerebrospinal fluid. This is so important because it's full of nutrients, full of oxygen, full of other nutrients that we need, that the brain needs to survive. So we're constantly turning that over. All right, so the cerebrospinal fluid is good for shock absorption, like I just said. Also good for buoyancy, like we just talked about. It floats the brain. It actually really reduces the weight of the brain by about 97% because it's just floating in this fluid, right? And then provides nutrients to the brain, as I just mentioned. Okay, so the locations of the CSF. This is a better image to show these different ventricles in the brain, but your brain has big cavities called ventricles, and that's where the majority of the cerebrospinal fluid is. But it's also in the subarachnoid space, so that's all around the outside of the brain. Okay, and then we have it right down the central canal of the spinal cord as well. Okay, so one pathology that's related to this is called hydrocephalus. Hydrocephalus is when we get too much water on the brain. And in kids, when those cranial bones haven't fused yet, this actually expands their whole skull. And you can actually see where this little guy's cranial bones are separating, right? And that fontanelle there, too much water in the brain expands that, can cause brain damage. If this happens in adults, you don't see that growth of the head because the bones are fused, but it can start pushing down on the brain and create too much pressure and cause damage. It's typically caused by this duct becoming blocked. So it doesn't circulate and you're making more, keep making more, but it's not draining like it's supposed to. Okay, here's some other things you don't need to know, but everybody always has questions about how it's caused. And so there's some causes right there. All right, and then lastly, the blood-brain barrier is an important protective piece to the brain. And as we talked about before, the glial cells that help us do this are those astrocytes, right? So it's kind of this almost oxymoron that there's no blood in the brain, right? When you look at an image of somebody this is a cool image, it's all blood vessels, right? You see that just incredibly dense amount of blood vessels in the brain, okay? But every single one of those blood vessels is surrounded by astrocytes like this. Okay, so all those blood vessels are surrounded by astrocytes. That's making a wall, a barrier between the neurons and the blood itself. So we have another layer of controlling what comes into the brain and what comes out. And that's what that blood-brain barrier does. All right, guys, that's all we got. See you soon.